Hello students, today I'll be solving most of the questions given by the board for the chapter name Reproduction in Lower and Higher Plants. So let's start with MCQs. The outer layer of pollen grain is thick and made up of complex non-biodegradable substance called as, and the options are A. Lignin, B. Cellulose, C. Pectin, D. Sporopollenin. And the right answer is D. Sporopollenin. Sporoderm is made up of, and these are the options. The right option is D. Exine and intine. Exine and intine collectively is called as sporoderm. The number of meiotic and mitotic division necessary for development of female gametophyte in angiosperm is. These are the options. And the right answer is D. 1 meiosis and 3 mitosis. You can see in this diagram, first megaspore mother cell undergoes meiosis and forms 4 haploid megaspore, which further undergoes 3 mitotic division to form female gametophyte. So 1 meiosis and this 3 consecutive mitotic division. This is mitotic division 1, 2 and 3. Identify the odd one with respect to pollinating agent. These are the options, and the right answer is B. Bottle brush. In bobag, kadam and sawsack, pollination is done by bats, that is Chiropterophilous. In bottle brush, pollination is done by birds. So this is the image of bottle brush. In vitro pollen germination and pollen tube elongation can be induced by, these are the 4 options, and the right answer is D. Sucrose. Cells incompatibility is found in flowers of plant. These are the options, and the right answer is C. Thea. So this is the image of Thea. Orogamy refers to entry of pollen tube through. These are the 4 options, and the right answer is C. Micropyle. You can see here, when pollen tube enters through micropyle, it is called as porogamy. Here, when pollen tube enters through chalaza, it is called as chalazogamy. In this case, when pollen tube enters through integument, it is called as mesogamy. Dash is an example of halobial endosperm. These are the 4 options, and the right answer is C. Asphodilus. Single shield shaped cotyledon in monocot seed is known as. These are the 4 options, and the right answer is B. Scutellum. So here, this is the scutellum. It is a single shield shaped cotyledon. Example of dicot endospermic seed is. These are the 4 options, and the right answer is A. Castor. Now very short answers. Here one more question. Why anther is called as tetrasporangiate structure? Answer is presence of 4 pollen sac in Dithecus anther. So here you can see this is Dithecus anther having 2 lobes, and each lobe consists of 2 pollen sac. So total there are 4 pollen sac, and that is why it is called as tetrasporangiate. At which stage pollen grains are shed from the anther in angiosperm? And the answer is bicelled structure. So this is 2-celled structure of pollen grain. What is hilum with respect to ovule? Answer: place of attachment of funiculus with main body of ovule. So this is the body, and the stalk of an ovule is called as funicle. The region where body is attached to funicle is called as hilum. What is protandry? Condition where androecium matures earlier the gynoecium. Name any one plant in which double fertilization was discovered. Answer is Fritillaria or Lilium. So this is Fritillaria, this is Lilium. Why fertilization process in angiosperm is called as double fertilization? Answer is both male gametes are used. Which is the most common type of endosperm in angiospermic families? Answer is nuclear type. What is the role of suspensor during the development of embryo? It pushes embryo into the endosperm. What is adventive polyembryony? Embryo develops from somatic cells or integments. Name the hormone produced by unfertilized ovary responsible for enlargement of ovary into fruit. Answer is indole 3-acetic acid. It's auxin. 2 marks question. Draw a well-labeled diagram of TS of anther. So for drawing the diagram, half mark, and any 3 labelings, half mark each. The labelings are your epidermis, endothecium, middle layer, tapetum, sporogenous tissue, and connective. Describe the structure of pollen grain. So this is the structure of pollen grain. It is single-celled, made up of single nucleus, consists of 2 layers, outer layer called as exine, inner layer called as intine, exine and intine collectively called as sporoderm, and this is the germ pore. When microspore mother cell undergoes meiosis, it gives rise to 4 haploid microspore, that is pollen grain. Draw a well-labeled diagram of male gametophyte of angiosperm. So this is the structure of male gametophyte. You can see here, this is the pollen tube, tube nucleus, and 2 non-motile male gametes. So this region constitute male gametophyte. Describe the structure of female gametophyte of angiosperm. So this is the structure of female gametophyte, also called as embryo sac. It is 7 cell structure and 8 nucleated. At the micropylar end are present 2 synergids and egg cell. 2 synergids plus egg cell is called as egg apparatus. At the chalazal end are present 3 antipodal cells, and this is 2 polar nuclei. When they fuse together, it forms secondary nucleus. Mention various adaptations for wind pollination. Flowers are small, inconspicuous, colorless, without nectar and fragrance, that is odor. The pollens are light in weight, dry, and produced in large numbers to increase chances of pollination considering wastage of pollen grain. Stigma is feathery to trap pollens carried by wind current. Stamens with long filament and versatile anther. Stamens and stigmas are exposed to air currents. Explain heterostyly and herkogamy with suitable example. Heterostyly, that is heteromorphy. Example is primula, that is primrose. There are 2 to 3 forms of flowers in which stigmas and anther are placed at different levels. So here in this picture you can see stigmas and anthers are at different levels. It prevents the pollens from reaching the stigmas and pollinating it. Herkogamy, it's a mechanical device to prevent self-pollination in a bisexual flower. In plants, natural physical barrier is present between two sex organs to avoid contact of pollen with stigma of the same flower. Example is Calotropis. It has pentagular stigma, is positioned above the level of anther. So this is the example Calotropis, and this is the pentagular stigma. Give the significance of double fertilization. It's a unique feature of angiosperm. It ensures that the parent plant invests a seed with a food store only if the egg is fertilized. The diploid zygote develops into an embryo, which develops into a new plant. The triploid pen, that is primary endosperm nucleus, develops into nutritive endosperm tissue. It restores the diploid condition by fusion of haploid male gamete with haploid female gamete through syngamy. It also helps to avoid polyembryony. Mention significance of fruit and seed formation. Fruit provide nourishment to the developing seeds. Fruits protect the seed in immature condition. Seeds serves as important propagating organs, units of plant. Seeds and fruit develop special devices for their dispersal and thus helps in the distribution of the species. Give an account of polyembryony. It is the development of more than one embryos inside the seed, first noticed by Leeuwenhoek in the seeds of Citrus genus. Occurrence of more than one embryo in a seed, which consequently result in the emergence of multiple seedlings. The additional embryos result from the differentiation and development of various paternal and zygotic tissue associated with the ovule of seed. In adventive polyembryony, embryo develops directly from the diploid cell of nucellus and integument. Example citrus. In cleavage polyembryony, zygote proembryo sometimes divides into many parts or units. Each unit then develops into an embryo. Polyembryony increases the chances of survival of the new plant. 3 marks question. Describe internal structure of anther. The structures are epidermis, endothecium, middle layer, tapetum. Epidermis is the outermost protective layer. Endothecium is subepidermal layer. Middle layer, inner to endothecium. Tapetum is the innermost nutritive layer of anther wall. Explain the development of male gametophyte in angiosperm. So let's see this by diagram. This is single-celled pollen grain. The nucleus divides and it becomes two-celled pollen grain. It consists of vegetative cell and generative cell. Generative cell divides by mitosis and forms two male gametes. Further, the intine comes out forming a pollen tube which consists of tube nucleus and two non-motile male gametes. So this region constitutes male gametophyte. Give an account of any two biotic agent for pollination along with their adaptation. Adaptation in Chiropterophilous flower. They are large, showy, and often brightly colored. The flowers produce sweet odor and have nectar glands. The stigma is rough due to presence of hair or sticky due to mucilaginous secretion. The pollen grains are spiny and surrounded by yellow sticky substance called pollen kit. Adaptation in Chiropterophilous flower. that is pollination done by bats. Flowers are dull colored with strong fragrance. They secrete abundant nectar. Flower produced large amount of edible pollen grains. Example is kadam tree, bobak tree, sosak tree, and these are the images. Kadam tree, Anthocephalus, bobak tree, Adansonia, sosak tree, Kigelia. Explain any two contrivances or outbreeding devices for pollination. Outbreeding devices avoids self-pollination and favors cross-pollination. First is unisexuality. Second is dichogamy. In unisexuality, plant bears either male flowers or female flowers. It is called as dioecium. As flowers are unisexual, self-pollination is not possible. Plant may be monoecious. Same plant produce male and female flowers. Example maize, or in dioecious, male and female plant are separate. Example mulberry and papaya. Dichogamy, anthers and stigma mature at different times in a bisexual flower. This prevents self-pollination. And it is of two types, protandry and protogyny. In protandry, androecium matures earlier than the gynoecium. Example, disflorate of sunflower. Protogyny, gynoecium matures earlier than the androecium. Example is gloriosa. Write a note on different types of endosperm in angiosperm. There are three types of endosperm on the basis of mode of development: nuclear type, cellular type, and halobial type. This is nuclear type. The primary endosperm nucleus undergo free nuclear mitotic division, means cell wall formation does not take place. So there are many nuclei. Then vacuole develops in the center, which pushes the nuclei towards the periphery, and in the end, the nuclei are accompanied by cell wall formation. So this is nuclear type. This is cellular type. Every cell divides to form two cells. Here there is nuclear division as well as cytokinesis, means nuclear division and cell wall formation takes place at the same time. And this is halobial type. Describe the development of dicot embryo in flowering plants. So let's see this with the help of diagram. Zygote divides to form two cells. At the micropilar end, there is large basal cell, also called as suspensor initial cell. Toward the chalazal end is present small terminal cell, that is embryonal initial cell. Basal cell divides to form suspensor. Embryonal initial divides to form 8 cell stage structure, that is octant stage. Here lower tiers of 4 cells of octant forms hypocotyl and radical. Higher tier of 4 cells of octant forms pumil and cotyledon. So here there will be 2 cotyledons. Then the cell of suspensor down, that is hypophysis, it forms part of radicle and root cap. Upper cells of the tier it divides to form heart-shaped structure of embryo, and further the cells divide and redivide to form horseshoe-shaped structure, that is embryo. Draw a well-labeled diagram of monocot seeds. So this is the structure of monocot seed. It's an endospermic seed. You can see the endosperm. This is the embryo, which consists of scutellum, that is cotyledon, radicle enclosed by coleorhiza, pumil enclosed by coleoptile. This is the seed coat, and this is the aleuron layer. explain various categories of apomixis. It is a phenomenon of formation of embryos through a sexual method of reproduction, and it is of recurrent apomixis, non-recurrent apomixis, and adventive embryony. 4 marks question. Describe the structure of anatropous ovule with the help of labeled diagram. So let's see this with the help of diagram. Let's start from the center. This is embryo sac, also called as female gametophyte. It is 7-cell structure and 8 nucleated. At the micropylar end, there are 2 synergids, an egg cell. Synergids and egg cell together is called as egg apparatus. At the chalazal end are present 3 antipodal cell, and this is the secondary nucleus. Embryo sac is further surrounded by nucellus, that is megasporangium. It is made up of diploid parenchyma cell. Nucellus in turn is surrounded by inner integument and outer integument. This is micropyle. It's a small opening between 2 integuments. This part is chalaza, from which arises integument. From micropyle to chalaza is the body of an ovule. The stalk of an ovule is called as funicle, and the region where body get attached to the funicle is called as hilum. Describe the development of female gametophyte of angiosperm with the help of diagram. So let's see with the help of diagram. Megaspore mother cell undergo meiosis and forms 4 haploid megaspore, of which this 3 degenerates. Now this single megaspore get nourished by nucellus, which further undergoes first mitotic division to form 2 nuclei, then second mitotic division to form 4 nuclei, ultimately third mitotic division to form 8 nuclei, and here the nuclei get rearranged and ultimately it forms female gametophyte, that is embryo sac. Give an account of various abiotic agencies used in pollination along with the adaptation for pollination. Abiotic agents, they are non-living, that is water and wind. Adaptation in anemophilous flower. Here pollination is done by wind. Adaptation in hydrophilous flower. When pollination done by water, these are the adaptations seen in hydrophilous flower. Flowers are small and inconspicuous. Perianth and other floral parts are unvetable. Pollen grains are long and unvetable due to presence of mucilage. Nectar and fragrance are lacking in flowers. Give an account of pollen-pistil interaction in details. It's interaction of pollen grains with sporophytic tissue, stigma. It begins with pollination and ends with fertilization. All the events from the deposition of pollen grain on stigma to the entry of pollen tube in the ovule are referred as pollen-pistil interaction. Describe the process of double fertilization in angiosperm and add a note on its significance. In double fertilization, fertilization takes place twice. First is syngamy, fusion of male gamete with female gamete to form zygote. Then triple fusion, second male gamete fuse with secondary nucleus to form pen, that is primary endosperm nucleus. It was discovered by S.G. Navasin in Fritillaria and Lilium. When the pollen tube passes through micropyle, it is sporogamy. When passes through chalaza, it is called as chalazogamy. When it passes through integument, it is called as mesogamy. Significance of double fertilization. It's a unique feature of angiosperm. Ensures that parent plant invests a seed with a food store only if the egg is fertilized. The diploid zygote develops into an embryo, which develops into a new plant. The triploid pen, that is primary endosperm nucleus, develops into nutritive endosperm tissue. It restores the diploid condition by fusion of haploid male gamete with haploid female gamete, that is syngamy. Also helps to avoid polyembryony.